Hello student, good afternoon. To continue the lecture, that is the life cycle of wuchereria bancrofti or filaria bancrofti. The further development of the microfilariae depends on their being sucked with blood by certain species of mosquitoes. In order to infect mosquitoes, there may be about 15 or more microfilariae per drops of blood. A high concentration of 100 or more per drop of blood is fatal to mosquitoes. Shortly after being digested by a mosquitoes, the microfilariae penetrates through the stomach wall and migrate to the breast muscles. Here the embryos lie lengthwise between the muscle fibers. The size of the body is reduced to half the original size length but grows several times as thick. It now changes into the sausage-shaped creature. The sausage-shaped larvae measures 1.5 to 2.0 in length and 20 to 30 micron in diameter. As these changes takes place, the larva molds twice and becomes infective. The formulation of infective stage takes place about 10 to 14 days and occurs best at 28 degree centigrade. A relative humidity 90%, digestive system, body cavity and genital organs also become well developed. This is called third stage larva. This infective stage reaches into the head of the mosquito and finally into the proboscis on about 14th day. There are several larvae coiled up the mosquito ready for infection in man. The complete development of the microfilariae in the mosquito is from the 10 to 20 days. Infection and adult stage in man. When the infected mosquito bites a man, the third stage larvae are deposited, usually a pair of skin near the site of puncture. Later on the larvae enter through puncture or penetrates through the skin. From here they reach the lymphatic vessels and settle down at same spot, that is inguinal, scrotal or abnormal lymphatics. The larvae grow into adults after a period of 5 to 18 months. Larvae becomes sexually mature and starts new generation of microfilariae. Pathogenesis and clinical symptoms. The location of the adult worms in the lymph glands and connective tissues indicates that they would induce inflammatory and immune responses, and indeed the pathogenesis is largely of this nature before patent microfilariae, that is before the microfilariae appears in the blood. The infection of symptomless then follows the acute stage marked by the acute inflammation of various parts of the lymphatic system, which becomes swollen or painful, accompanied by headache, nausea, urticaria, elephantoid, or filarial fever with temperature reaching 40 degrees centigrade, 104 degree Fahrenheit, and sweating. The inflammation of the lymphatic system manifests in itself in lymphangitis, lymphadenitis, inflammation of the lymph ducts and lymph nodes, respectively. Lymphoedema, chyluria, lymph in urine, making the urine milky in color, lymph, scrotum, and hydrocele, and lymph varies in which the lymph is blocked in the lymph ducts. If these conditions persist over a long period, usually for years, the lymphoedema becomes chronic, especially in the lower limbs, scrotum, vulva, breast, and arms with the become swollen and hard. There is fibrosis and hyperplasia of the tissues, and the skin thickness and shows folds and cracks in places, creating sites for secondary bacterial infection. Thus, the condition known as elephantiasis gradually develops. It is now known that elephantiasis does not arise from blockage of the lymph ducts by adult worms, but is the result of the allergic reactions to metabolic products of the worms over a long period. In fact, there are usually no microfilariae in the blood. When elephantiasis sets in Eastern Africa, particularly in Ethiopia, studies by Women and Price have shown in some areas where elephantiasis of the legs in endemic wuchereria bancrofti does not seem to occur, and so the conditions cannot be due to bancroftian filariasis. Price was able to show a link between the occurrence of elephantiasis and the type of alkaline volcanic soil common in Ethiopia. He therefore postulated that soluble silicates enter the body through wounds in the skin of the feet and legs, crystallize in the lymph nodes, inducing inflammatory reactions which ultimately contributed to the onset of elephantiasis. Also, Castellani 1969 believed that elephantiasis not directly caused by the wuchereria bancrofti, since even through filariae sites may kill the worms. Recurrent attacks of lymphangitis parasite he recognized may form elephantiasis and put forward the Castellani theory, which shows the elephantiasis nostralis found in the temperate zone and elephantiasis tropica found in the tropics and supposed to the caused by the filariae are essentially the same disease whose etiology is bacterial streptococcal. In the temperate zone, the bacterial infections is the primary cause, while in the tropics it may be the secondary cause. The filarial may be serving as an irritating agent, paving the way for bacterial infection. Okay, thank you. Remaining part we will be continue in next lecture.